Thank you very much. Thank you to Kerala State. Thank you to University of Kerala and all the people who have been involved in arranging this opportunity for us. Today I am not going to talk about the QARC reaction. I'm going to actually change a little bit and talk more scientifically about another click reaction which we find is actually very exciting. It's an intramolecular reaction and we call it ENIC reaction. I'll come back to what it is later on. But initially, I would just like to give a brief overview of why we obtained this Nobel Prize. So what does it take to become a click reaction, the ENIC reaction? But before we go into that, I would like to say that when you get a Nobel Prize, you are asked to have opinions about everything. In order to facilitate that message, I have 4 mantras here, which I think are the most important issues, probably worldwide. First of all, chemistry is everything. There's nothing around us that is not chemistry. When we are hungry, it's chemistry. When we feel pain, it's bradykinin. When we feel good, it's dopamine, or maybe it's oxytocin that create empathy towards our children. It's whip peptides when we feel hungry, and so on. So there's a lot of things that goes into understanding the chemistry world that we live in, and the way that is chemistry is the basis for development of life. Because chemistry is everything, because chemistry is our food, our cars, our computers, everything in there is made out of molecules that have been put together in an organized manner. Because of that, we need to have our youth concerned with chemistry from the very beginning. So from first class in the curriculum, we should include pleasurable learning of the 3-dimensional world of chemistry. Not strange formulas that is hard to understand, but actually what it looks like and why it reacts like it does. And then we should have research without constraint, because all disruptive discoveries comes out of research where we have results that are unexpected. If it was expected, somebody would have done it before. If it was expected, it would be very easy to do. If it's unexpected, and when we can observe it in our chemistry as something we do not plan, just happens like that. And if we are observant, we can actually make use of that information. In order to get that to work, we need funding without restraints. So that means funding by performance. If we perform very well for the last 3 years, short horizon, we should accept that as researchers. Short horizon, have we done a lot of work in the last 3 years and it resulted in good results, we could publish. We get some more money to continue the good process instead of having blue sky ideas, a lot of competition and so on, which is judged by people who don't have the capacity to actually judge what is applied for. So I think funding by performance is the best way to go forward in research. I share the prize with Caroline Bertozzi and Barry Sharpless, 2 very good friends of mine. We were in the US several times around 2000, where Barry was in for the Nobel Prize. I actually have 2 of his small vials with the chemicals that he won from his first Nobel Prize. So at Carlsberg Laboratory, I started working on polymers, all sorts of chemistries in order to perform combinatorial chemistry on solid support, make many compounds. And this is a unique platform where we have polymers that can be used for synthesis as well as bioassay. So we can do a lot of chemical reactions on these supports, and we can make combinatorial libraries of millions of compounds. And we can also do enzymatic reactions on these supports. So that's a very good starting point. And during all of this work, we of course came on to the QARC reaction by Christian Wenzel-Thorne, who was a PhD student at the time. But we also developed something that is called N-acylaminium ion reactions on solid support, and that was again a completely serendipitous observation. So the QARC chemistry came because Christian wanted to do isolation of acetylenes with acid chlorides, and we used this technology that we had developed in the lab with the aceto-acid chlorides. That should work very easily to get us these kind of compounds, but it didn't happen. Instead, we got the triazole with a fully active acid chloride. Completely unexpected, but Christian, he wanted to quit his PhD study. He said, this is not possible to do, I don't want to do it. But we looked into this reaction and it was very, very promising, and it did exactly what we were looking for with the solid phase combinatorial chemistry. So we took this reaction and investigated it with all the different functionalities you have in proteins, and then Christian could present it in 2001 in APS. What is it? Yeah, an alkyne is a bag full of electrons. These electrons is in the HOMO here, and this HOMO is looking for electron-deficient molecules, which could be, for example, a copper 1. Could also have been silver 1 or gold 1, but copper 1 has a special property of acting as a very good catalyst for these reactions. It sits on the electrons, and that acidifies the proton. It takes the place of the proton, and another copper 1 cation is coming in to remove the electrons from the alkyne, so it opens it up like a can of sardines. Get to the LUMO. This is the LUMO of the alkyne with the metals on it, and that fits exactly with the HOMO of the A side, which then creates the two new bonds and provide us with a triazole. First, we have the loss of one copper atom, then a second copper atom, and you get the triazole product. And this is why it's so extremely selective and efficient. So Christian was looking into developing a new set of chemistries which are completely orthogonal to this huge toolbox that we have in organic chemistry. And we were not the only one looking into that. Also Caroline Bertozzi, Barry Sharpless, and later on the TCO reaction in 2008 fits into this orthogonal little toolbox where chemical reactions in the little toolbox don't influence any chemistry in the big toolbox and vice versa. So that creates a lot of opportunities to build molecular robots or do structural control with click reactions. Here's a cell membrane, and we have used these kind of molecules that you see here. These kind of molecules, the targoplasines, which are the killers of these animals in the innate immune system. And we have looked into their activity, and we made these beta hairpins with triazoles, and then we try to open up the cell membrane the way that it works, this antibiotic. The content of the cell flows out, and you get the activity to work for you. This is what it looks like, the targoplasine, and we are replacing these two disulfide bonds with triazoles to see if that would work for us. And we try that on 4 different cell lines, and you can see one of the cell lines is completely resistant to targoplasine 1, whereas replacing the 2 disulfide bonds with our triazole gives a fully active peptide. So this is the background for the click reactions, and if I should say what it needs, what you need to become a click reaction, it is that you have a large delta G for reaction, at least 50 kilojoule per mole, to ensure that you have a quantitative chemical transformation at low concentrations. The click functional group should be inert to other reaction conditions, and also all other groups should be inert to the click reaction conditions. This is a key to click reactions. The click reaction should also preferably be readily installed independent of the molecular architecture. The click reaction should be selective, clean, and orthogonal at room temperature. At room temperature is very important. It should be stereo and radio selective as well as biocompatible. It should perform independent on the environment, including water and oxygen. It should be triggered preferably. If you can trigger it, you can control it either by, for example, light or by a catalyst. And then it should preferably facilitate it through templated coordination, and it should be enable synthesis of otherwise inaccessible molecular structures and properties. There's also a nice to have here, and these are just, the normal preparative nice to haves. But the need to haves are up there. So what I would like to go through today is these structures that you have here in the center by the enike reaction. This is our platform, combinatorial chemistry, assays to isolate active. We analyze them, we inject them in a mouse, and we can see the activity of a particular enzyme here in this foot. We can grow the cells on the beads, find actives, and analyze them and go there. I'll come more into this assay towards the end. So we were trying to install aldehydes on the N-terminal of a peptide in order to use them as electrophiles to build functionality, organic functionality into the peptides. And this was very, very difficult until we found out that we should just install a methyl group on the nitrogens, then everything would work. But then we started to investigate what is it that makes it difficult for using the normal peptides. And to our surprise, the aldehydes actually reacted with the amide bonds in the peptide backbone. So that could be immediately something new. And here is the aldehyde. Here is a small peptide with a C nucleophile sitting here on the site. So we release this aldehyde. It forms the hydroxylactam. That forms an extremely reactive N-acylaminium ion here. And this N-acylaminium ion is looking for electron density. It finds it right here on the tryptophan, and it closes the ring. And it does so with 100% stereoselectivity. So this is a very prominent new reaction, and we will now like to see if it can be used generally for all sorts of purposes. So the reason it works so well is that if we do molecular dynamics, we can see the aldehyde are very close to the amide bonds in the backbone, and it does a ring closure here. And then this nucleophile is situated right on top of it to do the next step of the reaction. So we would like to have a number of accesses to aldehyde building blocks, and here is shown a malonic ester synthesis. First we install the aldehyde here. We protect the aldehyde, and then we install a side chain, amino acid side chain here, and that is a building block for aldehyde introduction into peptides. We can also do this with phenylacetic ester synthesis, just installing the aldehyde and protecting it to give us this kind of building block. That's two methods. The third method is by oxidation of a diol. Protection here, oxidation of a diol, protection and oxidation in the other end, so we get a long chain carboxylic acid. We can also do an oxidation of a vinyl group to give an aldehyde here. And these are extremely good conditions for that reaction, which is necessary because we carry it out on solid support. Finally, we can use the amino acids themselves as a source of aldehydes by reducing this part to the aldehyde and connecting this via a carbonyl or carbamate to the peptide backbone. So we do first a reduction to the aldehyde, then we do the protection of the aldehyde. We do a N-terminal deprotection, and then we add this carbonyl pentafluorophenyl carbamate. Now, does these reactions really work as click reactions? And one of the requirements is that you have a quantitative reaction under mild conditions. And we can, with 10% TFA, that's very mild acidic conditions, we can get this reaction to go in these four cases here. Very easily, you can see a quantitative formation of just one product by HPLC. This is crude products of the resin. And here you see the NMR spectra, very clean reactions. Only one stereoisomer is formed. And that stereoisomer we can analyze by NOE, by NMR here. Proton 1 here has a good NOE, strong NOE to proton 3. So this is the absolute configuration of that molecule. And they are 100% stereoselective in all of these reactions. Here we have the aldehyde with the side chain and a lot of different substituents here on the tryptophanes. These substituents on the tryptophanes are introduced by synthesis, so a lot of them you can only get as a racemic mixture here. Our building block can be racemic here. So if we look at the next slide, we can see the result is 2 peaks because we have a non-resolved stereocenter, but the reaction is completely stereoselective. The same if we have these racemic tryptophanes, you can see we get 2 products, but these are formed in a completely diastereoselective manner. And that goes all the way through here. So we have a reaction that is completely stereoselective. It's happening in water with a little bit of TFA. That works well with the more reactive C nucleophiles, but as we reduce the reactivity, this doesn't happen so easily anymore. So here we treat with 10% TFA to form the hydroxylactam, and in order to get this to react with this weakly reactive C nucleophile, we need a stronger acid, and that's shown down here. Take this one, 10% sulfuric acid in acetic acid or 50% TFA in dichloromethane, and we get only 1 product. And it's actually important here to have first the weak acid treatment on the solid support and then the strong acid treatment after that. So we tried that with a number of different substituents here to see how does that vary the yield if we have different electron withdrawing or donating groups. And you can see in all cases we get a single product out of this reaction. So it's extremely selective and very nice reaction that only forms 1 stereoisomer. In this case, we had expected also to get some here, but because of the reduced reactivity, it really finds the place where there's most space. If we look, on the other hand, on this methoxy group here, we definitely see both products because here we have much more pushing of the electrons from the oxygen into the ring. So this side and this side are evenly reactive. And you can see here, this is the para substitution and this is the auto substitution due to steric reasons. This is smaller. If we take off the metal group, we get more space and we get a larger one with the auto substitution. We can also use polyaromatic systems like these ones to form a lot of rings here. And these reactions are also extremely selective. Here we form only 1 product in each case, shown here. Here we are a bit surprised that we only get the product here and not here. And you can see we form a single product with this structure. Here we have nitrogen, sulfur, and oxygen. So we have an R group, which can be either a proton or isobutyl or benzyl, different types of substituents here. And then we have this X as a nucleophile. We get these kind of structures with different ring sizes, and you can see in all cases we get a good product, except for the long chain here. This is a lysine side chain, and this is a very short chain, a serine chain. We don't get any product there. We get a complex mixture. But all the other cases, it worked fantastically, and you can see here the product that we are forming, and it's a festivitas of heterocyclic systems that we are getting out of this. If there's no nucleophile sitting in the side chain, the N-acylaminium ion will actually find the next amide bond and do a double condensation to give these interesting structures. We can also combine this with other organic reactions like the Suzuki reaction. Here we carry out either before or after a Suzuki reaction here at this aromatic residue. all sorts of substituents in these two positions. And we use diphenylphosphine, bis-diphenylphosphine ferrocene dichloride in order to do this reaction. It's a very efficient catalyst for the Suzuki reaction. And it gives us quantitative yields in this first step. The rest of it is also quantitative, so we get very high yields, as you can see here. And this is also shown by the HPLCs of each of these crude products. And here we can install any kind of functionality on the aromatic systems. So a very powerful way to generate these heterocycles. As you have noticed, this ring here has mostly been a 5-membered ring. We would like to know, can we expand that? And we made these longer chains. You can see now we get very good yields, except here where we have n equals 2. So if we have a long chain here, just like we saw before, we don't get a good yield. But here, 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 and here, we get very good yields. So we can make a 6-6 system or 6-7 system, but we cannot make it larger there. So here's examples where we have done this with different nucleophiles, and you can see in all cases we get a quantitative transformation. The vinyl group is a very good source of aldehydes, and it is a very good source of aldehydes because we have the osmium tetroxide sodium periodate oxidation of these, and in the presence of DAPCO, and I must say we have tried every single base in the universe. DAPCO is the only one that is really, really superior here. DAPCO gives us 100% yield of the aldehyde in no time. This is DAPCO. And then we get the hydroxylactam, the iminium ion, and it can condensate here. We have done here with a variety of different substituents on the phenyl group. And here you will see the result. You can see we're just moving methyl groups around here, putting in phenols, making these polyaromatic compounds as well. So it works really well with the vinyl groups as well. So very good source for this reaction. Very easy to install. Here's the vinyl benzenes. We do the oxidation with osmium tetroxide, dihydroxylation, cleavage with the periodate in the presence of DAPCO, and then TFA 50%, and we get the ring closure to this heterocycle. Very interesting compounds, actually. Here's another one in which we are using an O nucleophile instead of a C nucleophile. We can also make sort of a diketopiperazine in this manner using acrylic amides, making the oxidation, getting the aldehyde, and getting condensation. Completely new heterocyclic ring system here. This can be done with proline or with N-ethyl amino acids, which promote the cis orientation to give the hydroxylactam. This is installed here, and you can see we have now formed a locked molecule where we have an amino group here to continue with and a carboxylic acid here. And we wanted to use that for something, and what we focused on was the synthesis of enkephalin analogs, which would be highly active on the mu receptor. And this is allyl glycine installed. We have an O nucleophile here, so we can make this kind of cycle, and then we can continue the synthesis and make this compound with a very locked conformation here. Very active agonist. Over here, we do it differently. We synthesize the peptide first, and then we can actually install this moiety completely selectively after the synthesis. So this is another way to actually do that. Get a very selective reaction. So you can see this is also very pure according to HPLC. The INSIG reaction is another version of the ENIC reaction in which we are using the amino acids. We have turned them around and made an aldehyde from the amino acid, and we use a carbonyl group to connect two amines here. This gives us this kind of product in the reaction. So the aldehyde has reacted with this one and with the nucleophile and give us 2 new rings. Almost completely stereoselectively this time. It can be done with a lot of variety of structure, as you can see. And this is the summary of what the different structures can look like. One of the interesting things, this is sort of how that works. You can see one main product in all cases, not quite as selective as the enite reaction, though. So here is a typical reaction. We start out with this dimethoxyphenylalanine. We couple this PFP carbamate onto the amine, and then we do the release of the aldehyde. It reacts with the amide over here, and then we form this new ring and this new ring, and we have a compound. This can actually oxidize, and if we add a little bit of DDQ, we can do this very rapidly to get these kind of interesting positively charged anions, or cations, and these are highly fluorescent. And not only are they highly fluorescent, they are also selective for binding a certain ligands. Here is a phosphate buffer at pH 7. Here is water. And this one is water, pH 7. Here is phosphate, pH 7. And you can see the bead simply shines up a lot. So we can also do a dual reaction here. First, we do one reaction on the amino group out here at the end with this one. And that forms, here on the side chain, that forms this imidazolone. This imidazolone is now acting as a nucleophile here on a second reaction with a similar compound like this one. We are reacting this amine on this carbonyl group, and then we do the release of the aldehyde to get this compound initially, and that then form these ones on treatment with TFA, completely new heterocyclic ring systems made from the diversity of peptides. So extremely useful. And this is what it looks like in the HPLC. So here's a last example where we are using this reaction to form a ligand for CB1, CB2 receptors. The reaction is first an amide formation here, an imide formation here with this compound with a bromide here. Then we do the amide formation here at this carboxylate to form the amide. We do a reduction, a selective reduction on one of these 2 carbonyl groups in the dimembered ring to give the hydroxylactam. That forms the iminium that reacts with this C nucleophile and give us that structure here. Now we are adding an amine by amidolysis of this aromatic bromide with palladium, and we get this product here. And why are we then synthesizing that product? Well, if we look at this product and fit it into the receptor of CB1 and CB2, it fits exactly into this pocket. And what we would like to do here is to separate the dimerization of this receptor and get the response that is selective for homodimerization and not have the response which is heterodimerization with the HT receptor, which is responsible for the psychotropic effects. So in summary, we can make all of these different ring sizes. We have a very stable structure also around the ring system, and it can be totally combinatorialized. 5, 6, 7-membered rings here, 5, 6-membered rings here, CNSO nucleophiles in this position. We have this hydrogen bond, and this stereochemistry is 100% trans. You also can use the L or the D or the LD or DL all the way through this, so you have a lot of combinatorial possibilities. And this is just a small fraction of the number of templates that we have actually. the number of templates that we have actually synthesized. And if I was a pharmaceutical company, I would look a little bit into this because there's a lot of interesting compounds here. So we would like to see, can we use this combinatorially? Now we have developed the method, and here is the method, which is screening of GPCRs with large one-bead, one-compound libraries. And the assays are based on cells that adhere to this adhesion molecule, which in itself was developed by combinatorial synthesis. We got the best one, which is this one. And that is then attached to the bead together with one of these heterocyclic ligands. When we do that, we can grow the cells in a monolayer. It interacts with the compound on the bead and gives us a signal that we can read, and we can then decipher the compound on the bead to find out what it is. So the way it works is that we have an expression here of a EYFP together with the melanocortin 4 receptor. And when this is activated, this receptor is activated, we get a fluorescent signal in the cell. So when we interact the G-coupled protein with the ligand on the bead, it sends a signal to the nucleus, and we start seeing fluorescent cells. And here you see the example from the library where some cells are lighting up while other beads don't light up at all. Yeah, so this is a melanocortin receptor in the membrane, and we have been looking at this because it's involved in obesity. So we would like here to actually see if we can control obesity with small molecules. We have previously developed a very good assay with macrocyclic peptide ligands, finding very good selectivities. But can we also screen this in a small molecule combinatorial library like this one? Different substituents everywhere. And we have 34,500 compounds in the library that we then treat with the cells. And you can see here a bead where the cells are lighting up. Here's another one. And here. And what we see is general for these ones is that we have a phenyl group out here or benzyl group out in this position. So we captured a lot of active beads, and we used mass spectrometry to identify what is the compound that is active. We can see the major peak here is simply this heterocyclic portion of the molecule, so it's quite easy to decipher the structure. So we got the active compound. Here's one of them, but if we look at it more closely, you can see here is an indole and a benzyl, indole and a benzyl, indole and a phenyl, indole and a phenyl. And this is the most reactive one, in which the methyl group sticks up here, whereas this one is less active, is this one, and so on. So there is actually quite good activity, submicromolar EC50s of these compounds. I'd now like to just conclude by saying we have 4 types of click reactions here. The QARC reaction that we got the Nobel Prize for, the spark reaction that Caroline got it for, this TCO reaction, which came too late. It came in 2008. And this reaction here, which I think is a very, very useful reaction for a lot of future chemistry. So with that, I would like to thank not only you, but in particular all the people who have been involved in this work, Sarah, Mads, Thomas Nielsen in particular, and Frederik Dines, also Jürgen Bayer, who was the first one to observe the aldehyde formation. No, sorry, that was actually Jörg Rademann who did the first observation of this reaction with the aldehyde and the backbone of the peptide. And then I would like to thank my family, in particular Phaedra Saint-Hilaire, who has been very proliferative in discussions, scientific discussions about all these issues along the way, and my family for accepting late hours in the laboratory. And thank you to you for your attention.